Good evening, everybody. It's my true pleasure to welcome everybody back to a 28th season of our Sea Secrets 2023 series. We'll have 6 lectures. I'm Rani Avissar, the Dean of the School, and I want to thank our sponsor for this lecture series, which is obviously extremely important for us. Without that support, we could not, in fact, maintain this activity. So in particular, I'd like to thank again the Shepard Broad Foundation, Bill Galway, Cheryl Gold, the KB Life Enhancement Forum, the Key Biscayne Community Foundation, John McCann Family Foundation, Gail Nansen in honor of Russell F. Nansen, Nicole and Myron Wang, and Southern Glazers Wine and Spirit. We have a tradition before each one of our speakers. We try to present to you one of our distinguished alumni. And tonight it is my great pleasure to introduce to you Liv Williamson, that many of you already know. She is a member of the Coral Reef team. She got her Ph.D. here with us in 2022 and got her undergrad from Barnard College, right in Columbia, if I remember that correctly. So she is a senior research associate with us now, and Liv tests intervention strategies to increase survival and fitness of reef building corals. She seeks to optimize outcomes of reef restoration efforts using managed breeding and microbiome manipulations to enhance coral resilience during early life stage. Aside from research, Liv shares her passion for the underwater world with the next generation of environmental stewards through science, communication, outreach, and citizen science. And she has also an amazing gentle dog that, we share in the park when I meet her regularly with my own little dog, and we have time to speak about science and other things. So, Liv, all yours. Thank you.
Good evening. I'm going to present to you my quick little 3-minute thesis talk, which won last year's 2022 3-minute thesis competition here at the University of Miami and won the People's Choice Award at the Florida statewide 3-minute thesis competition. Okay. I was really hoping to gesticulate out here. All right, I'll gesticulate back here instead. All right. I want you to think of an animal that impacts your life every day. You might think of the dog or cat you share your home with, or the cows whose meat or dairy you consume, or maybe the bees that help our crops and gardens grow. Chances are you are not thinking about corals. But just offshore, Florida has the world's 3rd largest coral reef. So for residents here and for 500 million people around the world, corals are integral to our way of life. Corals are ecosystem engineers, building reefs that provide habitat for the seafood we eat, produce ingredients for our medicines, protect our beaches and coastal properties from flooding and erosion, support tourism and recreation, and hold immense cultural value. Sadly, in the past few decades, we have lost half of the world's reefs and the critical ecosystem services they once provided. Climate change is the primary culprit. Healthy corals depend on tiny algae that live in their tissues in a symbiotic relationship and help them survive and grow. But extreme marine heat waves trigger a process called bleaching, where corals lose their algae, causing starvation and death if they don't get them back. If coral reefs are to survive climate change, interventions are urgently needed to help them take the heat. One way corals might adapt to warming oceans is by changing which algae they host. Previous studies found that a type of algae called durastinium allows some corals to withstand marine heat waves and avoid bleaching. With my PhD, I tested a scalable way to deliver these heat-tolerant durastinium algae to lots of corals by doing so very early in life. Baby corals are being bred for reef restoration efforts here in Florida, here at UM in fact, and worldwide. They're very small, so we can work with hundreds, even thousands of them at once, and most are not born with algae of their own. So I asked, can we increase the heat tolerance of baby corals by giving them durastinium algae? And does this approach scale for reef restoration? To answer this, I reared several endangered species, including elkhorn, brain, and star corals, and provided them with different types of algae sourced from both adult coral colonies and laboratory cultures. During their first few months of life, I seeded thousands of baby corals with Durastinium algae and found that most formed healthy, stable partnerships. Importantly, baby corals hosting Durastinium algae survived twice as long under high temperature as those hosting other algae. My research showed that in the face of rapidly warming oceans, actively supplying baby corals with Durastinium algae before we plant them onto the reef can help boost their heat tolerance. Every single baby coral that avoids bleaching and survives on the reef long term will add brand new genetic diversity and climate readiness to coral populations that so badly need it, enabling coral reefs to continue sustaining coastal communities like ours into the future. Thank you.
Thanks, Liv, and again, congratulations on winning this award. Scholarships help us, recruit and kind of educate also students like Liv. And if any of you is interested to know about our scholarship programs and wants or is interested to contribute, you can contact Jennifer and speak with her about that. It is now my true pleasure to introduce the speaker of tonight, Professor David Kaplan. As I mentioned before, Professor David Kaplan is a member of the faculty at Tufts University. He is the Stern Family Endowed Professor of Engineering and a professor in the Department of Biomedical Engineering, which I understand he has established at Tufts about 20-something years ago and run for about 20 years. I can tell you as an administrator that has been running an organization for almost 14 years, all my respect to Professor Kaplan for managing that for 20 years. It's a great achievement on its own. But really, Professor Kaplan is here tonight to speak with us about his field of expertise, which is on biopolymer engineering, tissue engineering, regenerative medicine, and cellular agriculture. He has published over 1,000 peer-reviewed papers. So for the scientists of us, that gives us an idea of the productivity. He has a team, I understand, of about 45 postdocs and graduate students, okay? And so that generates about 50 papers a year, okay? It's just one paper per student, no big deal, right, per year. But that's an amazing productivity. He's also the editor-in-chief of ACS Biomaterials Science and Engineering, and he serves on many editorial boards and programs for journals and universities. He directed the NIH P41 Tissue Engineering Resource Center that involves Tufts University and Columbia University for 15 years. His lab has been responsible for over 150 patents issued or allowed in numerous startup companies. He has also received several awards for his research and teaching and was elected Fellow of the American Institute of Medical and Biological Engineering and to the National Academy of Engineering. Dr. Kaplan recently received funding from the US Department of Agriculture to establish the first national center in the US dedicated to research in cellular agriculture. Professor Kaplan, it's a true pleasure to have you at C-Secrets. Thank you.
So first of all, good evening, right? It's evening now. Thank you, first of all, everyone for showing up in the evening, donating some of your time to come listen. I really appreciate it. I know everyone has busy lives. Also, to those of you online, I appreciate that as well. For me, it's a joy getting out of the cold weather in the Northeast in Boston and spending a few days here. So my great appreciation to the dean, obviously, and the organizers, Tatiana particularly, for all the help, the logistics to get me here, at least in one piece. We'll see if I get home in one piece tomorrow, but that's to debate. What I wanted to do today is talk about sort of the most recent area of our focused research in the team that you heard about, and that is this field of cellular agriculture. So today we're going to talk about this general title. I called it Meat and Fish Foods of the Future. So really what we're going to talk about is what your food is going to look like on the table maybe 10 years from now, maybe 20 years from now. You can debate that if you will, but this is where I think some of the future foods will come in terms of alternatives to current sources. So there are different terms that people use for this. People talk about growing food without the animal, if you will. That's the common thing we use. Or ultimately, from the scientific and technological standpoint, it's tissue engineering. Putting the right cells, the right scaffold or material together under the right conditions to create something you would want to eat and you would enjoy eating, right? Those are the keys there. How are we going to do that in a useful way? So really, this is farming without animals or fishing without fish. This is really the theme I want to talk about today. And I have 3 different parts of this I want to cover. First, just a broad overview so you know really what I'm talking about here. What does it mean when I talk about cellular agriculture? And why should you prepare for your future dinner table? In the middle, I want to talk about some science. I'll show you some examples of the science we have done in this field and why at least we're excited about what that means for the future. And at the end, I'll talk about some of my harebrained ideas on where I think this is going to go and what the impact could be in a much more substantive way as well. Because I see this as the future on how we're going to feed the planet. So let's try this again. Why do I keep missing? Let me try the arrows. No. Okay. Any help here? It's frozen again. I can't. It's a very sharp talk tonight. I'll use the arrows. That might make it easier. Okay, good. Okay. All right. So to start, these are things you may have already seen out there, right? You may see things like the Impossible Burger, or if you go to KFC, you see chicken nuggets. These are plant-derived products. These are not cellular agriculture. These are materials derived solely from plants. Another example is just this mung bean protein used as a scrambled egg. These are all plant-derived products. So just keep that in mind. This is really not what we're going to address tonight. And those examples I just showed you, there are many, many more, come from this kind of process. Very briefly, working left to right, you're taking some existing plant. Could be corn, could be a mung bean, could be a soybean, and you're chopping it up, crushing it, mushing it, baking it, and then extruding it somehow through a machine to make a product that you then make and look like something you're used to eating. Great, right? That is very heavily processed material, oftentimes heavily salted, other ingredients, extenders to make it palatable, tasty, etc. But yet the cost is pretty good. So they're competitive with current foods. So over the last 3 to 5 years, you've seen an incredible influx of these kinds of foods. In every restaurant, you can buy something like this. In the supermarkets, you find this, and they have become quite popular. The problem is the consumer excitement has now started to wax and wane, right? It started high, and now it's starting to decrease. Because these foods don't really fully recapture the things you want in your food, things you're used to that you want, the taste, the flavor, the chew, whatever it might be. And so these help because it points to how hungry, pardon the pun, consumers are for new alternative foods, but yet these get part the way there, but not all the way there. And so how are we going to get all the way there? And this is what we're talking about tonight together, cellular agriculture. Why do I think this is the case? Because essentially you're going to eventually make foods exactly the same way in terms of composition that you consume today, but you're going to do it free of animal utility in the process, or free of the fish in the process. And so briefly, just to walk you through this, I'll give you a lot more details. We take an animal, could be a cow, could be a fish. We do a muscle or a fat biopsy, which is take a small sample of tissue. And out of that, we isolate stem cells from those animals. And then we expand the stem cells to make lots of those cells. And then we expand them again, and we expand them again. And we keep expanding them, because you need a lot of cells to make the foods everyone likes to consume. And as we do that in the right kinds of systems in the middle there, we then design a biomaterial housing, a scaffold, a support structure onto which those cells will sit, grow, and differentiate into muscle or fat or connective tissue, the things that make up the food you like to eat. And when we're done, in principle on the right-hand side, we can make the foods out of this process that look exactly or very much like the foods you're used to eating, with the same flavor, texture, taste, and importantly, nutrition and safety. So this is the mandate. This is the goal for this topic tonight, right? And this is real, okay? You're going to see products coming out this year available in some restaurants based on this process. There's already one product in Singapore, and the first approvals from the USDA occurred in this country just a few months ago. So these are coming, not at the scale yet, just as early prototypes so people can start to get used to them. All right, so first of all, why do we care? Why should I waste my lab's resources and all of my passion and time on this? Why is it so important? It's important for many different reasons. The first one is what you all know about, right? Just in November, we passed 8 billion people on this planet. We cannot continue to feed people the kinds of protein, the kinds of meats, the kinds of enriched foods they want today at the rate we've been doing it. It's just not a sustainable approach to doing this, whether it's the fish from the ocean or whether it's farm animals on the farm. And this population, hopefully political issues aside, will continue to grow, right? And therefore, we're going to have more and more demand. And you see this all across the globe, not just in the US and not just in Europe. You see it in China, India, and elsewhere. So this is the future. And the other motivation here is on this slide. And this is why I get the most amazing students who get involved in this research that I've had in my entire career at Tufts. Students are concerned about population, we just talked about that, right? But also just the general issue of sustainability. How do we make food without damaging the planet, right? Whether it's water resources, the marine environment, and so on. How do we improve animal welfare, right? If you look at things like antibiotic use on the planet, I think it's about 80% of all the use is just to expose farm animals to the antibiotics, to keep infections down and keep them safe. So food quality becomes a question. Food security becomes a question. You saw this with COVID, right? The hotspots for COVID started out in meat processing plants because this is where a lot of propagation happened. So food safety and food security become really, really important here. And then finally, food equity. How do we make food and high quality food available to everyone and avoid things like food deserts and so on. So there are many motivations for what we're talking about tonight. They're summarized here, but they all come to bear, and this is where students from different interests come into the field and contribute to what we're doing. And the other thing to keep in mind for cellular agriculture, it is not a one-stop shop, right? You need expertise from many, many different disciplines. You can see this around the curve here, right? Whether it's consumer acceptance to nutrition, to cell biology, to molecular biology, to everything else in between. They all have to work together. That's one reason why we have a large lab, right? We can't do this independently. You need to make sure you have all the skill sets working at the same time. So we'll come back to this towards the end when I talk about education a bit, but just have the sense that you need lots of different perspectives to make this work. All right, so I put this slide together just because, and this is focused on fish because I knew we were here tonight, right? So to honor the process, but this could apply to many different animals in the middle, right? But we can take something like the fish tonight. And if you look at the challenges, and I think I'm speaking to the choir, or at least I assume I am, so if I'm not, go ahead and throw a piece of paper at me, but we're faced with depleted fish stocks. We're harvesting fish to chop it up and feed back to fish in aquaculture. Nano and microplastics have become an issue. Toxicity is a problem, and obviously costs for fish continue to go up. That's assuming even the fish are high-quality fish that you want to eat today because there's so many exposures like this, right? So with cellular agriculture coming into the fore, right, if we can grow fish the way I just described to you, harvest the cells, grow up the cells, create the right textures, shapes, et cetera, in principle, then this process becomes much more controlled, much safer. Think about pharma, producing drugs in a confined environment. That's what we're talking about, really. Producing foods to the level where it's very consistent, high quality, safe, nutritious, et cetera. The other advantage, and I'll talk about this in a minute, is what I call tunable nutrition. And we've shown this in our work already. I'll show you one example where you're not sort of left to whatever the animal is going to eat, metabolize, process, and provide in its muscle or its fat. You can control the process at the cell level. So we can tune the composition, which means you can tune the nutrition, and you can improve the quality of what you're going to eat. I talk about discoveries. I'll show you some examples just in the past 3 years in our lab, and there are many others out there as well, where we've taken the concern for cost, which is way too high right now, to make this a viable technology. And we've reduced the media cost, the biggest culprit here, by 90% just by some simple discoveries we and others are making in the field. And then the last big challenge is scaling. You know, if I didn't put a slide in, but people talk about if you take every bioreactor, every container to grow cells that you would get from the pharmaceutical industry, from the brewing industry, from the enzyme production industry, you would still be like at 1% to 10% of the total volume you need, you know, to grow the amount of food we're going to need. So there's huge challenges ahead. Don't be fooled by what I'm telling you tonight. What I'm saying is the science and the technology is coming along really well, and you're starting to see companies come into the fore for the scaling and other issues, but it's going to take a bit of time. But we are very, very optimistic. And one way you can measure optimism is what you see on this slide. And this is now a bit old, but this is just the influx of new companies in the past few years that are in this space, not just cell ag, but also alternative proteins and so on. So this is a booming economy. This is the next wave, right? Now it's slowed down a bit with the current sort of slowdown in the overall world economy, but this will bounce back as well. So the growth of new companies and even the growth of existing large food companies towards cell ag is very measurable. They are either developing their own segments of their teams to focus on this, or they're investing heavily in the startups in the space to see which ones succeed and which ones they're going to invest in further. So this is coming. It's here already. The question is, how fast will it impact in terms of providing these alternatives to you as a consumer, for example? All right, so how does this work? So here's some examples from our lab. This is sort of phase 2 now, unless I'm talking too much, what I'm wanting to do usually. So I want to talk you through some examples from the lab, as I mentioned. So here's one. This is a couple of years ago. At Tufts, we have a school of veterinary medicine. So the students went out. We were able to biopsy a cow, brought the sample back to the lab, which is what you see on the left, isolated the stem cells, and then propagated them, which is at the bottom left. And what you see in the panel on the right are those cells growing in the laboratory, so in vitro, free of the animal now, and continuing to grow until at the bottom right, you see this beautiful fibrous bundle. And that's what you would see in a muscle, as if you took it from the actual animal itself. So the point of this slide is very simple. You can harvest cells, you can grow them up, and you can cause them to become muscle in a dish, and they look just as good as the muscle you would get from the living animal itself. So that's one bit of science that's very important to continue to grow for every species, right? You can't do it just for cows. We need all the different species that people like to consume. And so that's great. So now we have a muscle, which is on the right-hand side here, which we can achieve a certain scale. But it's nothing close to what you see on the left-hand side. Nothing even remotely close yet. We'll talk later about how we're getting there, but keep in mind the gap between what I'm talking about so far and where we need to get to. You can do the same thing for fat. Okay, these are examples of fat from both mouse fat and also porcine or pig fat. And the point on the left-hand side is you see that kind of opalescence coating on the bottom of a plastic dish. Those are just pure extended fat cells, full of fat, ready to go and be harvested. And that's what we do on the right-hand side. You can see right below the red pipette, there's a glob of white fat there. That's the collected fat from the surface, bound together with an enzyme, and now you have three-dimensional animal fat grown without the animal completely. Okay, so it makes the point, we know how to do this at the scale we're talking about on these past few slides. The other really important thing is what's on this slide, when I mentioned this earlier. you can take those fat cells, for example, and change what you feed them as they expand to make lots of fat. So they're not confined to what the animal tells it to do through its metabolic profile. It's defined by what you as the scientist give to those fat cells. So first of all, in the middle, you see two green displayed examples of the fat globules on a dish. And you can barely tell the difference. One is from the real mouse, and one is grown in the lab. The point is we can make the size, the shapes, the things you want to give you that kind of texture. And the longer we grow the cells, the more fat the cells accumulate. So you can make a decision on how much fat you want in those cells to go into your food that you're going to make through cellular agriculture. More importantly, I would argue on the right-hand side is what I would call composition. So if we look at something like omega-3 fatty acids, which are the healthier fatty acids, we can dial in how much or what the ratio is of those fatty acids, again, based on what we feed the cells during their cultivation and expansion. So why do I think it's important? Oh God, we froze again. I'm sorry. Hold on. Yikes. Oop, there we go. Okay, good. We unfroze. So why do I think it's important? If you look at the left-hand side, right, inputs to a cell growing in the lab, you have control of the nutrients, the fatty acids, whatever supplements you want, which means the output, the cells themselves will be fortified in a way you cannot possibly achieve with animals today, living animals or fish or so on. And so the cells win time and again in terms of tuning and controlling the nutritional composition. And these are things you'll see growing in the future as well. We'll talk about more of these examples as we go. The other way you could do this is with fish. Okay, so this is something we've done at the lab. I even went out on deep sea fishing out of Gloucester, Massachusetts, right? That's our lab doing this. You don't see me in the picture because I was the one who got seasick, or one of the two who got seasick, so I'm back in the cabin. But we caught fish, right? We brought the fish back to the lab. Same process I just described. Muscle biopsies, fat biopsies, et cetera. Isolate the cells, grow them up. And now you're faced with a slightly different problem because cows and chickens and pigs, they've been well studied in the past, right? So there's a lot of data on what to look for to make sure you have the right cell and the cell is kind of from what the lineage is and so on. With a lot of fish, we don't have that luxury. There's not a lot of databases out there and a lot of detail about what to look for. So we had to go back in and we had to sequence the genome of this particular isolated muscle stem cell that we thought was the right cell. We wanted to make sure, and we could validate that it was. Once we had that, we were very fortunate. I'm not showing you all the data, but we just kept growing and growing the cells until they spontaneously immortalized. And for those of you not familiar with what that means I can grow the cells here, I can grow them here, I can grow them up there, I can grow them everywhere, like Dr. Seuss would say, right? But you could grow them forever, right? They will be able to be grown forever. Why is that so important in the field? That's because that means I could ship those cells to every one of you, and you could grow your mackerel cells, which is what these were, in your kitchen, and you get the same product in every kitchen, right? Without that, you would have to go back to the ocean, harvest another mackerel, and isolate cells again. And that defeats the whole purpose here. So this way, the mackerel can live their lives in the ocean. Obviously, people will still catch mackerel, that's not the point, but you can grow the food and make it mackerel-like cells and tissues at the same time. And so we were fortunate. Within 1 year of that discovery process, the technology was licensed to a startup company. The fancy name is Wanda Fish for obvious reasons, but we won't get into there. Okay, so the other thing we do a lot of strange things, I call it in the lab, strange things that I think have very important meaning for what we're talking about tonight. And one is we're very active in the field of insect cell culture as well. And not a lot of labs think this is a viable path, but we do. And so if you just look at this set of conditions here, we could isolate insect cells. And insect cells don't need fancy growth ingredients. They don't need expensive ingredients. They don't need a lot of what we call process control to make sure they behave properly. And so because of that, the cost to grow these cells becomes very small compared to fish cells and mammalian cells. So you're already way ahead of the game if you can bring costs down so precipitously. And that's what we're doing by carrying out this kind of technology. It's already well known insect cells are nutritionally very analogous to anything you would eat today, if not better. And number 3 on the list, you can find insects that have flavors that mimic almost anything you've ever eaten. It's important to realize that the majority of the population on this planet actually eats insects every single day of their lives, right? Other than the US and Europe, everyone else does. When I've traveled in Korea and China and Southeast Asia, I've eaten insects, not routinely, but occasionally, just to see if I could deal with it, and I did. So, you know. So I'm not saying you're going to have insect cell-derived meats on your table anytime soon, but I do think on the right-hand side here, as fish feed, to get away from harvesting fish to feed back to fish in aquaculture, this is a winner, right? It has to be a winner in terms of nutrition, cost, and so on. I think as livestock feed and pet food as well, this is also going to be a winner. So we will see. And if you can think about the millions of insect species on this planet, we know almost nothing about the cells from each of these, how easy they are to propagate. Think about insects living in almost any environment, ultra cold, ultra warm, in the marine environment or not. So you could have cells that really are very sort of matched to whatever niche you want to grow the cells at. But eventually, I do think these will find their way into human food. 2 quick examples. One is here. This is, and the term we've coined here is entomoculture, but using insect cells to generate cultivated meat. And I already talked about the reasons why. We work on 3 different species right now. One is crickets. I didn't put that up here. One is a tobacco hornworm caterpillar, because I have a colleague in biology, Barry Trimmer, who studies these. And you can see these beautiful rib muscles that come from the insect as an example of how you could grow highly organized muscle tissue. And the fruit fly, just because the genetics are well known, so it gives us a template to work from. But I just put one slide of data on the insects just to make the point in a colorimetric outcome. So if you look at the 2 left-hand panels of images, and at the top, this is where we set up insect cells and set up mammalian cells side by side. They're all green. That means they're healthy, happy, doing well. On the left-hand side, the C2, C12s, those are mammalian muscle cells. And the ones to the right, the long name I won't describe, is a muscle cell from an insect. And what we did is we set these up, we closed the lid, and we ignored them for a month. Didn't do a thing to them, right? And if you look and follow down the days, a month later, the bottom left, everything's red or purple. It means they're all dead. And the other one is all green and beautiful chunks of cells. They're all, all living and doing well. Those are the insect cells. So if you think about cost, there's nothing to do, right? I mean, there is, but very little to do compared to the mammalian cells. So this is going to be a game changer. Again, whether it's going to be 3 years or 30 years, I can't tell you that. But certainly, in terms of the science, the underlying science and motivation here, this has to be something useful in the future, whether it's for fish feed, pet food, and so on. All right. The other really important factor I've mentioned a few times is just the cost to do this, which is way too high. And most of that cost, when people do life cycle analysis and related issues, it always comes down to the growth media. So the fancy ingredients you need to feed these cells so they'll grow and differentiate and make the muscle and make the fat. And there's a long list on the left-hand side, but the 2 most egregious components, one is serum, which comes from cows, as you may know. This is bovine serum albumin. And the other are just these very expensive proteins. We call these growth factors that the cells need for instructions on when to grow and when to differentiate. And so we and others are very active in trying to understand how to develop substitutes that are more cost effective. So I'll show you a couple of slides on this, and underpinning all this is we're very active with colleagues in our consortium to develop modeling tools as well. So in the future we can pre-predict what to feed cells of different kinds to get the best outcomes at the lower cost. So, for example, if we want to remove serum, which is a huge cost, it's also animal-derived, and if we're trying to get away from growing animals, then it doesn't make sense to use serum as well. And you can see in the graph here, this is our success with recombinant albumin. So this can be produced in plants or bacteria, still pretty costly, but better than bovine-derived serum. And if you look at the black line there, this is looking at cell doubling, so how many times cells keep propagating against days on the x-axis. And you see the black line, that's the sort of the cocktail at the five-star restaurant. That's giving them every cost you can, as much serum as you can, making them fat and happy. That's what you get. But you can see if we replace all that serum, that very expensive serum, with recombinant albumin, you see the blue and the red lines do really pretty well. They're not so bad at all. So that's one example of an immediate win here. And if you don't use any serum or any of the recombinant albumin, the bottom line peters off very quickly. The cells die off. Okay, and the important thing here on the right-hand side, we can still plot the cells, and we can see they still propagate well, and they still eventually differentiate into muscle cells. That's what the pictures show you on the right. All right, so albumin is better, or is a good substitute for serum, but it's still expensive. So now we can get rid of the albumin and go with just a plant extract. So think of a low-cost waste product from the agriculture industry, and now you can see the curves on the left. Again, they're a little bit lower than the Holy Grail there, but they're doing really, really well. And now we've dropped costs precipitously. And again, the cells do really well still. And then the last part here is we talked about the fish cells going through immortalization spontaneously. The bovine cells are more difficult in this case. And so we have to use genetic tools to override the control of cell cycle to get these cells to be immortalized. And you can see that with the pink line here, where the cells just keep doubling and doubling and doubling without a problem, whereas the non-immortalized cells kind of end up around 50 doublings, and then they start to lose their vitality. Importantly, you'll notice the little insert there may be hard to read, but the average doubling time for these cells is about 15 hours. That's pretty good for these cells to grow up in culture and scale and so on. So the bottom line for all this of substituting growth factors and serum, the costly stuff, is summarized on this slide here, right? Just in a couple of years in our lab, and again, this is going on in other labs as well, we've gone from, in principle, almost $200 a liter to grow cells. A non-starter if you're going to grow food for people, right? But now we're already down to the right-hand side at basically a tenth, almost a tenth of where we started. And that's with very, very little effort so far. So the science, again, is pushing this forward very quickly. I think we'll continue to see immense decreases in costs going forward. And I think that's where now the cost comparisons will start to come in, and we'll have the ability to start to make foods that will be compatible in terms of cost for consumers. All right, and the last part on this is just in growing fish, since again we're here, there are so many drivers that make fish cell culture for the cellular agriculture approach a really big winner as well, whether it's the oxygen control, whether it's pH variability, temperature, and so on, and ultimately the nutrition you all know about. This, again, will be a driver into the future as well. So time will tell, but that's the estimate. Okay, so we talked a lot about cost here, okay, and now we're going to move into the future. The future is how do we not only deal with cost, because we have a good progress there, but how do we deal with scaling? And how do we deal with making a steak as opposed to a hamburger? All the challenges ahead, okay? So, so kind of put on your Doctor Who hat, which is one of my favorite shows to watch with my daughter. And first of all, let's just talk about scaling in general. A lot of people are working on using 3D printing in the field to try and achieve these goals. We use it at small scale. I'm not sure you'll ever see this at large scale. It's just going to be very expensive to do. At least that's my assumption. The other way people look at this is to borrow from the pharmaceutical industry and use these large steel tank reactors. These are called stir tank reactors. This is how most of the pharmaceuticals are made today, by growing bacteria and fungi, isolating the products, and making the drugs that you use or may use, right? Again, I think this is going to be good for a certain level of scaling, but I think it's going to be really challenging to scale at the level we're going to need. So you'll see a lot of this. It'll be really good, but to eventually see this as common alternative foods that you could buy in the supermarket, I'm not sure you'll get there with this method. So in our group, we have 3 or 4 different, what I call out-of-the-box solutions. I'm going to only show you one tonight, and it's prefaced with this slide, which is thinking about foods of the future, you have to rethink everything, okay? You can't be bound by existing protocols. And if you're going to make ground beef, that's not as challenging. But if you're going to make, fillets of fish or a steak, you really have to think about cells, texture, density, and these kinds of challenges. And so what we came up with and we're working very hard on is what we call textile engineering-based food production. So a very different strategy here. And the idea is very simple. I'll just walk you through this in 30 seconds. At the heart of this, you take an edible fiber or yarn, and you grow your cells, your muscle cells on there, or you grow your fat cells on there. And cells like that because they're in direct contact with the media. So they get access to oxygen, glucose, and the other things they need. So there's no limits in terms of diffusion limitations to get ingredients to large, thick slabs of tissues you're trying to grow. It's a huge challenge, and the cost goes up when you do that. So here we bypass the whole physical limitations that exist. We just grow things out of fiber. And then after that, you don't care what the cells do. They can even die because it doesn't matter. Just like when you buy food, the cells are no longer living when you buy raw meat, right? Or raw fish. And so here we just take and go through traditional textile engineering. Yarning, weaving, matting, laminating, you name it, you can do any of that and create 3D food textures. And here, if you twist and compress, you get the textures you would expect from a steak or something like this. So this is one way of a number of novel ways we see this that could become scalable, right? You could see factories of the future where you're just growing cells along yarns, and then those go into downstream processes as well. We also have programs through NASA. Oops, we're froze again. Try again. There we go. So we competed for what's called the Deep Space Food Challenge. How are you going to grow food in long-term space missions? So we come back to our favorites, the insects, right? They don't care what you do to them almost. So now you can bring a little powder of insects with you. You can bring the nutrients in powder form, so it's lightweight. You have a oven, a little bioreactor carried on board. You put the ingredients in, you let it sort of grow, and then it bops down to another chamber where it's cooked, and on you go. So food on demand in a confined environment. You can imagine this also in confined spaces. You could be on a submarine, or you could be in a sort of remote environment as well. So with the right cells and the right ingredients, this could all be sort of a powdered ingredient setup where that's all you would need to do this. All right, so that's one level of the future, a little bit different, and we like it because it's different. The other is really different, and this is sort of our vision for really where this field is going to go, perhaps in the longer term or not, right? So one thing you can think about that you could never do before is actually make food or meats or so on from endangered species or from species that have never been domesticated before. And you can do this because you don't need to harm the animals anymore, right? You just need a small biopsy. You never touch the animal again. And so this opens up the door for really novel foods that perhaps we've never tasted before, but we don't even know if we'll like them or not. But this is one way to think about the future. And if you really think about how narrow our food tastes are, I mean, we use a lot of spices and other things, but the core foods, it's chicken, beef, pork, a couple of fish, and that's about it, right? There's others, I know, but the bulk of it, right? So the future may be that there may be hundreds or thousands of variants you can consume. We don't know a lot about them, but there actually are startup companies doing this already. The most sort of prominent one you may know is called Vow Foods. They're based out of Australia, and they have been developing their first foods of non-traditional domesticated animals. So we'll see what that holds, but I think there's a lot of potential there. Just got to check on time. Okay, I'll speed up to finish. The other is therapeutic foods. I think the future here, we talked about how you can feed the cells what you want. You can also engineer the foods in different ways, and I'll show you one example in a second. So you can start to generate the cells that are going to go into the food that already have all your medications in them, or already have the right nutrient profiles. And you order online, and you get those foods for your particular needs at your particular time in your life, and you live a better life because of it. Nothing to prevent that from happening. We just have to scale and get it there. And this gets into personalized foods. You may have preferences based on your genetic profile, your metabolomics, and this can all be done to match your needs. Just like you're getting the right drug to take care of the right ailment, you get the right food to give you the best metabolic outcome that you want. Nothing to prevent this. And then you can make food fun, right? Which it already is, but you can make it even more fun. Because ultimately, everything I'm talking about, you can do in the kitchen, right? There's nothing to prevent that. You just have to buy the right ingredients. And so think of a bread machine, but now you're going to grow your food. So it could be a fun thing. You could 3D print the food and get the kids involved and so on, right? So that's the future we'll all face. We'll see where we, how quickly we get there. So I want to show you one example of therapeutic food or improving food. And very briefly, because I know I'm losing the rest of my time, this goes back to eating a lot of red meat, okay, which is summarized here. The sort of the take-home message when you eat a lot of red meat, in the center there, what happens? You get a lot of oxidative reactions. And particularly, that leads to a lot of challenges in terms of disrupting your normal metabolic functions. And there's been correlates of this with heart disease, colorectal cancer. That's all published work, not from our lab, that's other labs. So the suggestion is if you can deal with that oxidative damage to proteins and lipids, you could then improve the healthiness of the meats you would eat. And so that's what we did. This we published a few years ago. And you can see in brief, this is what we call synthetic biology. So we basically took the pathway to make vitamin A, like in a carrot, the orange color in the carrot, which is an antioxidant, and we imported that pathway into our bovine muscle cells. So think of a hybrid muscle cell from a cow with a carrot in there, right? That's kind of the image I want to leave you with, right? But it's real, okay? And so that's the genetic engineering we went through, so that's something to think about. And what does it look like? You can see it on the slide here. For time, I'll just point out the cells actually turn a little bit orange, not too bad, but a little bit orange, right? So you really know it's working quite well. And ultimately, the data all there says, in fact, the cells did better in terms of antioxidant protection, meaning they should be more shelf stable, right? So less food spoilage, allowed to be used longer in your refrigerator or when you buy it from the market. And number two, lower amount of oxidative damage, so they should be healthier when you eat them as well. So you can see the summary there. So we see this as really therapeutically beneficial, right? If you have inflammatory diseases in your gut, consuming these foods would in principle improve your outcomes on a regular food consumption basis. The protection was even better with the cooked food, meaning the antioxidant level stayed higher even upon thermal heating these materials. And you can let your brain go. Think of all the possible pathways you can import in these cells. And we're working on some of those as well today. So for time, we work on lots of the biomaterial components, the scaffolding to house the cells. These have to be edible. A lot of these come from the marine environment as well, so we're thinking about the chitosands, the alginates, and so on. So lots of beneficial points there. And we do a lot of nutrition screening and so on with everything we grow in the lab. We just recently did our first flavor analysis. We had approval to do human flavor assessments of the foods we're growing, and then we'll do edible consumption coming up as well. So I want to close with just two slides. The first one is this, just to mention, I focused on science today, right? But ultimately, education plays a huge role in what we're doing. So I'm very proud of the fact Tufts is really leading the way in educational resources for this field. Everything we've developed, we put online and other schools use now. So we have lots of programs. We even have undergraduate minors in cellular agriculture now. We have graduate degrees in cellular agriculture and more coming as we speak. So we want to expand those programs. We have obviously lots of research going on. I mentioned a little bit. We have strong collaborations with other academic labs as well as with industry in this space to move things forward. And we obviously publish our work and also provide patents for folks to use. We were very fortunate, the Dean mentioned this, the USDA awarded us a $10 million grant to establish what they call a National Institute for Cellular Agriculture about a year ago. And it's a constellation of schools. You can see them listed there. It's led by Tufts. Our main partners are Virginia Tech, UC Davis, as well as contributions from MIT, Virginia State, and UMass Boston. And the teams are amazing. They all work together towards the common goals I talked about briefly today. We have consortiums with industry through our university now. We have special projects run by industry as well, and obviously other grants and nonprofit support in this space. There's still not enough public funding in this space. We need a huge influx, almost when you see things like the electric car or precious metal recovery. We need that level of action here to make a difference. The field will grow, but not at the rate we need it to grow to catch up with the demands. All right, let me close with the most important slide, and that is my CellAg students on the left. You can see them there in various activities. Just a wonderful bunch. We've already graduated 3 full PhDs in CellAg. We're the first lab to do that, and they're already out there doing great things. We have too many colleagues and collaborations to go through all of these. I just figured since we're here, I'd mention we collaborate very actively with Bigelow Labs up in Maine for lobster breeding, so we can isolate cells from there. Obviously, UMass Boston is very big in aquaculture as well from different perspectives, so we work with them. WandaFish I mentioned as one of the spinoffs. And if you're not familiar, BioFin in the middle there is a startup from a former alum at Tufts in the aquaculture space as well. So lots of sort of marine-related activities that have started up in this space. So let me stop there. I just want to thank again the group here for having me down for this enjoyable opportunity to talk to you, and also the warm weather. And thank you for coming out tonight. And I'd love to answer some questions if you have any. Thank you very much.
Thank you so much, Dr. Kaplan. We have a couple of volunteers that are going to walk around the auditorium. So if you have a question, please raise your hand, and they'll come with a microphone so that everybody can hear.
Hi, well, this is really new to me, and it is fascinating, and thank you so much. But since it's new, I've got to ask a basic question. You're talking about the cells, you're feeding them the serum or the albumin to, so that they grow. And you get to the point, you get to that immortalization process. Okay, what makes the cells take on that immortalization process? Is it just enough food? And as you add additives to these cells, is it a possibility you're going to add something that's going to stop that immortalization process?
So great questions. So immortalization is very variable. There are many different spots in the genome where a change can happen that drives cell cycle to continue. You could go too far, right? You can go into a cancerous mode as well. So you want to make sure you don't. So there are safeguards to pursue all that. So once you have an immortalized cell, you have to keep track of it throughout the process. So just like any cultivars for plants or other things, you'll have these cells, they'll be stored. For example, when we develop our cells and our immortalized cells, we provide them to clearinghouses like Carafast or ATCC, so anybody can use them, right? The whole idea is to get them out there and be used. But we still have to track them because you want to make sure that they're not changing further to go down the dark side, right? So that is just like any cell used in pharma manufacturing, in beer brewing, in bread making. You just track the cells, and you have all the tools, analytical tools, to make sure they're fine. So that's standard, right? Could you feed something to the cells and they stop being immortalized? Absolutely. You have to, again, that's the science. You have to understand the system before you would ever use it in a food, and that's what's going on now. So yes, safety is paramount to go with the quality and so on. Yes, great question. Thank you.
Two very quick questions. At the very beginning, you mentioned COVID came from a meat processing plant. I thought it came from bats. How do I know? I don't know. So if you can explain that quickly. But the most important question is, when will we have rack of lamb like this?
I'll put on my project, I don't know. No, what I meant by the hotspots, the COVID propagation among the workers in the plant happened at meat processing plants, not the origins. Sorry if that was not clear. In terms of when, I don't know. I don't like to predict. I'm like I said, I know you'll see products in restaurants this year. That is for a fact. They're not going to be in the supermarket yet. That's going to take years, I think. So I think you'll start to see consumers having the opportunity to sample expensive early prototypes to see how they like them. And then we'll see where it goes. But hopefully 5 to 10 years from now, definitely you'll see it at a more regular basis, and then it'll grow from there. So it'll be an alternative to try for now and an alternative going forward for quite some time. Yes?
I have a question, in fact. Yeah, if nobody in the audience is asking. There were, a little bit of genetic manipulations of vegetables that has created a lot of media attentions, okay, and Frankenfood and all the problems that are generated from that. Do you anticipate similar reaction from the public about this kind of food?
Again, fantastic question. The answer is yes and no. We anticipate every possible reaction. Okay, that's a given. What we do is the fundamental science, and we believe we need to look at both non-GMO, non-genetically modified organisms, as well as genetically modified. I believe the rate at which you'll see these foods, your rack of lamb on the shelf, will be much, much faster if GMO is allowed. I think most people would agree with that. So the question is, what do consumers want, not what I care, right? So I care about the science. And ultimately, it'll be up to all of you what you're willing to buy. I can tell you the first approved cell ag product, which was Upside Foods, just 2 months ago, I think now in the US, through the USDA. They got approval for both non-GMO chicken and GMO chicken. I don't know which one they're going to manufacture and provide to consumers first, but they know they need both eventually. And it also is very country specific or region specific, right? The US is much more open to alternatives, I would say, than many parts of the world. Europe is pretty close to it. I was surprised China said no to GMO, so you don't know. Singapore, I'm not sure where they stand. So I think we'll see different variants. What I do want to emphasize, though, is the newer genetic tools. For those of you who may be geneticists or molecular biologists here, when we start to use things like CRISPR and related tools, it changes the ballgame in terms of regulatory landscape because you're not leaving any trace of any manipulation in the cell. And so it's going to be this gray area, is it GMO or not? And it'll all be in the labeling, and then consumers will make their own decision. So we want to provide all the alternatives, and consumers can make their own decision at that point.
Good evening. Let's see if I can state this question understandably. In terms of nutrient efficiency, do you think you'll be able to beat the animal model in terms of the nutrients required to grow your proteins?
I think if we don't, we're wasting our time, I guess is my answer. I mean, if I understand your question, right, if you think about a cow feeding off hay and how much ends up in meat, it's pitiful, right? So the answer is absolutely, we should far exceed in terms of efficiency. And we know that already. If we grow our cells in a reactor, we can track all the metabolites and optimize the process, almost in real time at this point. So in principle, we should be orders of magnitude better than the living animal, to be demonstrated, but that's the goal.
Time for two more questions, okay.
Thank you for a very eye-opening and stimulating talk. I'm wondering if insects are so much easier to have the final product than the fish and the bovine, whether you would consider some of the, number one, not temperate, but tropical, which have a much faster life cycle and grow at higher temperatures much better, and the crustaceans, which have much shorter cell lifetimes and, in fact, total lifetimes, where the cells may grow by themselves much faster, shrimp, lobster, a series of seafood, rather than the much more evolved fish. This might be useful.
Yeah, thank you. So first of all, the idea of the insects and getting tropical versus Arctic and so on, that's all on the table. We just haven't had enough hands and time and resources to get to all that, but that's the goal. One of the goals for the lab is to create a repository for all these cells that anyone can use to grow these things. So it'll take time, but we want to do exactly that, to see where you want to grow these under what conditions and so on. So stay tuned for that one. In terms of crustacea, shrimp, lobster, crab, There are already probably a dozen companies out there that reported, I don't know how real because they don't tell you the details, that they have isolated cells that are growing shrimp and lobster and crab tissue. Shiak Meats in Singapore has perfected the lobster, for example, according to their website. If you go on there, there's a couple of shrimp companies and so on. So there's definitely progress there. It's hard to know exactly where they're at because, again, companies don't share their details. But we are looking at those same species because we want to do it in our own hands so we get proper life cycle analysis. So, but some of these are very, very challenging to grow. We don't yet have all the recipes that work every time. So it takes a lot of effort to figure out, why are the cells not growing? What stage of the animal to isolate from? What muscle do we isolate from? How do you deal with contaminants that come from a marine environment that aren't present in a land environment, that you have to get rid of to avoid contaminating your culture? So the list is long. I made it sound really simple and easy tonight, but it's not, okay? But we are certainly working towards crab, shrimp, and lobster in our own lab, for sure.
Last question.
Yes, I had a question about the fact that cheeses and yogurts, etc., are produced with microbes. I wondered what role, if any, do the microbes play in the preparation of this food?
Yeah, another great question. So there are different ways microbes can or do play roles. So one very simple one is we talked about those expensive growth factors, right? They're a great way, we call this alt protein now is the catchy term, right? But great way to engineer microbes to crank out lots of those growth factors that are reasonably inexpensive to then feed to the cells, right? There are some labs working on what we call co-cultures, which is combining microbial cultures with these kinds of cells we're talking about tonight. That's a whole nother science, right? Because you have to find the sweet spot where both want to cohabitate and not destroy each other. But that's a very growing, active area that I think should bear a lot of fruit down the road. And then the last place they play a really big role in the future, in my mind, is when you grow large quantities of these cells that we talked about tonight. There's a lot of metabolic waste that microbes can take in and convert to useful materials. So looking at the whole cycle here becomes really important. So they're going to play a pivotal role, bacteria, fungi, and so on. So stay tuned there as well.
Thank you so much again, Dr. Kaplan. For those of you, or if you have friends that would like to see this presentation and has not been able to attend, it will be available on our YouTube channel and posted to Facebook. And then our next presentation in the series, that is on March 6th, is going to be online only and will be on climate action as a prescriptions for health from Professor Aaron Bernstein from Harvard. Thank you very much. Have a great evening.